iClicker. Hello, good morning everybody on Zoom. We're getting there. Okay, I got my long questions. Hello. Okay, let me share my screen before I forget to do that with my Zoom people. Oh, y'all, it looks like my hair's going crazy, eh? Okay, good morning. Let's start our class. Okay, we'll minimize and minimize. And attendance, we can start that. And then we minimize this. No. Okay, there we go. So attendance is up and rolling if you so choose. And let me open up my chat. Okay. All right, switch it up.
With chromatography separation, how do you tell when to switch off the tubes? Is it just an eye test?
It's all clear liquids.
Exactly. So like, how do you?
So you count drops, or you have little microcentrifuge tubes that have a 1 mil mark. And I'm going to collect every 1 mil. Yeah.
So it's a math thing. Every 1 mil you do.
But sometimes it's a bigger column, so sometimes 1 every 5 mils.
Okay.
It depends on the column.
All right. We did it in organic, but we had like slight difference in color a little bit, and we can do it.
Yeah, usually no. Usually that's what we would joke about. That all we did in lab is mix clear liquids.
Yeah. Okay. So that was my big question. Was like, I don't know how they.
No, you don't. You don't know. So usually when you're designing a new purification scheme, you do it smaller increments. So maybe you do it every half a mil, right? And then you test your samples, and you see, wow, I had this range of 10 samples that my protein was in. Well, how about instead of collecting every 5 mils, I collect every 1 mil, and so now I only have 5 fractions with my protein of interest, right? So you can adjust it.
But how do you know the rate that they'll all go through the column?
You have to try.
You just have to.
You trial and error. Mm-hmm.
And then you just.
And then you write it down.
And you see the mixture, and then you see, do I have a mixture of something in it, or do I have?
So then you have to run it on a gel.
Okay. Okay.
And then you'll see, okay, well, I have 1 band in these 5 lanes.
So like to figure out, for a standard experiment, for us to say if this much liquid or this much time, it's someone that went and like measured all the different times and came out with what specific time was for that thing. Somebody
did some work before you came through.
Yeah, all right.
That's exactly right. You got it. You got it. Okay. Here we go. I want to join my class. Oh yeah, y'all don't forget, I'm sorry, I didn't say it. I was chit-chatting. I have, my fault. We have some long clicker questions. If y'all didn't get one, long clicker questions. Oh, I didn't post them to Moodle. Oh, y'all. Let me go post them to Moodle real quick. I'll have 2 minutes. Okay. But we're not going to work on them today. I'm giving them to you so you can work on them and bring them back to class next time. But I would really like to put them on Moodle for you. So y'all just give me a second and I'm going to get that on Moodle. 6. Get my shareable link. Okay. This is in the wrong class on Moodle. That'll make a difference. I never got any. No, I'm posting them right now. I'm posting them right now. My bad. I got caught up this morning, and I'm posting them right now. URL. So this is chapter 6, long eye clicker questions. My bad, y'all. Now it's in a weird place. I just got to move it. Oh, I have the chapter 5 one's in a weird place too. That's so sad. Okay. All right, so chapter 6 is up. There we go. Okay. Y'all should be able to access them on Moodle now. I apologize. It's my fault. Oh, you need a handout thing before you sit down. One of those. Yay, okay. They're on. All right, so please check in with iClicker attendance, and you need a form. Okay, how we doing? Happy Monday, everybody. Oh no, I didn't put a joke in my son's lunchbox today. Oh my God, I'm gonna be in so much trouble when I get home. Yeah, y'all gotta, but I had to save this joke to tell him at the end of the day because it's such a good joke. Can y'all help me out? All right. Was he telling y'all something in class today?
Yeah.
I don't know the herbivore. What's the herbivore one?
I went to the vegetarian group today. This girl, she said she swore she recognized it, but I don't think I've ever seen her before.
Oh, y'all, I put the wrong link. Hold on, wait. I did put the wrong link. Hold on. I wondered why it was automatically anyone with the link. Here we go. Fixing it right now. Thank y'all for telling me. Okay, let's see. Chapter 6, iClicker questions. I'm going to change this one. Edit settings. Okay. All right, it should be fixed. Adela, can you check for me, please, to see if I did it correctly? All right. So I want to go over something real quick on the long iClicker questions before we get caught up. So this is going to be for tomorrow, for Wednesday. It's good now. Thank you. Awesome. Okay, this is going to be for tomorrow. But I realize in those of you who took the handout copy, it's in black and white. So I want to point something out to you. And it would be great if I put the display on. I'm used to it already being on. All right, so for those of you who are sitting in class, if you look up at the screen when it finally loads, what you're going to see in the very first question is where you have that alignment of the human alpha, beta, and myoglobin genes. That those boxes are in red, right, because they correspond to the different helices, right? So the first helix is the A helix, then it's the B, then the C, right? And so in the question, they're going to ask you about E7, which means that it is the E helix and it's residue number 7. and they're going to ask you about the F helix. The F helix is in, I'm sorry, about F8. F8 is the residue that's in the F helix, and it is the 8th residue. Okay, so just make sure that you know that, because it's kind of hard to see in the printout copy. The blue didn't print too well, so just do realize that. And then when you're looking at these, if you want to know about what's being conserved and what's not, there are 3 different things that you're looking for. It could be where there's nothing. See how here there's nothing there? So that means that it's not conserved. Or there could be an asterisk like this, and that means it is highly conserved. So see how all these L's are the same, L, L, L? Or you could have 2 dots, which means it's not, look at the next lane, S, T, and S. It's conserved, but it's not perfectly conserved. Okay, so it has a high degree of homology, but it's not perfectly conserved. Okay, so those are your little reminders so that you know how to answer that one. And then I think I wanted to tell you something about the next one. Was it the next one? Where's my other page? Let me see. No? That's weird. Let me see. Oh no, and the other ones, oh, the other thing I wanted to tell y'all, if you look at number 3 and you look at number 5, right? These go along with the animations. I thought the animations were really, really nice for this chapter. And so I thought they would be a really good tool. So I built some questions around the animations. You don't necessarily have to have watched the animation and then go and answer it right away, but it would help. It would definitely help to like watch the animation, then come and try to answer the questions. Okay? And the animations are on Moodle. And they're on the online textbook as well, if you have the online textbook. If you can't access them, let me know, but I also recorded them in the lecture. So you should be good. Okay, am I finished taking the poll? Yeah, I'm gonna stop it when I go and start our iClicker questions. So let's see. I want the other one. Here we go. All right. So how was section 2? All about hemoglobin, myoglobin. It was kind of pretty content rich in this section, right? We learned a lot of new concepts in there. So I think we did question 3. Didn't we do question 3? Did we do question 3? Let me see. I'm full screen. Okay. So select the statement that is false. We did. Okay, good. We did. All right. So then 3 answer. Okay, question 4. Now, why is it that it's not fitting inside the window anymore? I don't understand. That's okay. All right. So let me stop our attendance poll. Now this one, I want as a short answer. So I want you to select the terms. Now this goes back to section 1 because we have a few left that we didn't catch on Friday. So select the terms that apply to the role of genomic caretaker proteins, right? Remember there are 4 different classifications. So look and tell me which one of these are genomic caretaker proteins. And when you list your answer, go 1, 2, 3, right? Okay. There will be more than one. Where is this iClicker question? Did it not send to you? What do you mean, Hannah? Yes, this is chapter 6. Remember, genomic caretaker proteins are an example of one of our proteins, one of our protein groups. It's taking a long time to upload. Okay. Well, you have the question here, so even if you can't click in on iClicker, it's okay. So this isn't a long iClicker question, Hannah. This is a short one. This isn't on the extra sheet. So I'll give y'all 10 more seconds. Y'all need 20 seconds. Oh no, I don't have these to print. I never do these to print. Okay, y'all ready? Here we go. Let's see what we got. All right, so hopefully you gave a couple of different, oh good. You gave a couple of different ones. It's not all of them. So what you should have said, oh yeah, and what role do the others refer to? I forgot about that. I forgot I did that. All right, so genomic caretaker. What does that mean, genomic caretaker, when we say that? So 3, 5, 6, 7, 8. There's 3. Ooh. Yeah. 3. Oh, y'all want to include more than what's in there. Y'all, 3. So when we talk about genomic caretaker, right? You have to think about what's required to maintain the genome. So you want to make sure that you don't have any mistakes, right? You need to fix your mistakes. And what else do you need to do? Replication. We need to make sure that that genome goes from one generation to the next generation, right? So what are some things that are involved in replication? Topoisomerase, we've already talked about, right? Topoisomerase is one that's going to release those supercoils so that we can replicate our DNA. DNA polymerase will copy it, right? Rec-A protein, we said, was a single-stranded DNA protein so that it keeps those strands apart. DNA repair proteins, because we want to make sure that we don't have any problems. And then 8 kind of encompasses them all, involved in replication and repair. So why not 1? 1 is involved, 1, 2, and 4 are involved in gene expression. So in transcription, right? That's not genomic caretaker. That's a different function. So yes, they are involved in, these proteins do work with the genome, but they are not caretaker proteins. Okay? So the others work in gene expression. Taylor, does that make sense? It's hard when I don't see the head nod of the people in Zoom. That's really difficult for me. Yeah.
So in the book it says a second major class of genomic caretaker proteins involved in gene expression. So those are involved
In caretaker proteins in DNA maintenance.
Right.
Yeah. So you have DNA maintenance. So both gene expression and DNA maintenance are caretaker, but there are 2 different functions within caretaking. Yeah, maybe I wasn't clear about that. I just said caretaker. Okay, I just said caretaker. So within genomic caretaker, we have 2 like subcategories. One of those subcategories is DNA maintenance, and the other subcategory is gene expression. So the Rec-A protein, the question on Zoom is, can you explain the Rec-A protein? So the Rec-A protein just binds to single-stranded DNA, and it keeps that single-stranded DNA separated. So you got to think about DNA replication, right? If as soon as you unzipped the DNA, it rezipped, you wouldn't be able to replicate it. So it just keeps it from rezipping, if that makes sense. So you good? Okay. All right. So I should have said caretaker proteins in DNA maintenance. All right. So how about this one? Structural proteins function as the blank for cells. This one's going to be multiple choice, as well as for tissues and organs. This one shouldn't take you more than a minute. Okay, so you got 5 seconds, 6. Okay, so what do they function as? Y'all should have said B, architectural framework. So when we talk about this, why did I have to say in this question, they function as framework for cells and framework for tissues and organs? Why did I have to make that separation? Do proteins that function as the skeleton of a cell also function in the extracellular matrix? No, they're different proteins. They're different. They are both structural proteins, yes, but they are different proteins. So now I'm going to ask you, there are multiple types of proteins. That's right, there are multiple types of proteins. So tell me, what are these cytoskeletal structural proteins? There are 2 major ones. Now, cyto means what? Before I do this, it's going to be short answer. But cyto means what? Y'all know this. Cell. Cyto means cell. So it's the cell's skeletal structure. So inside the cell. So I'm asking about the proteins that are inside the cell. We just said they're different for inside and outside. So what are the ones for inside? Huh? I'm trying to put my zoom on this one. Oh, there we go. All right, y'all ready? No? Y'all need another 30 seconds? Okay, y'all ready? Y'all extra 30. Okay, so what did we say? Actin and tubulin. Actin and tubulin. Tubulin and actin. Now, this is also true. Microtubules, microfilaments. Oh, did I? Wait, tubulin, actin, tubulin, actin. I think I got them all. Okay. So y'all are right. It's actin and tubulin. When we say these protein names, what are we referring to? An individual monomer. But these monomers assemble together to form filaments, right? Or long strands inside of the cell. Filaments. So microfilaments and microtubules. Yep. So Grace, did I miss you on the on here? Microfilaments, microtubules. Yeah, I got it. Okay. And so it's true, you could pick either one, but I want you to know the difference between using the words actin and tubulin versus using microfilaments, microtubules, thin filaments, things like that. So whenever you're talking about the filaments, you're talking about the monomers that have self-assembled together in order to make a polymer. Okay? All right. So actin and tubulin, that's what we said. All right. So now this protein not only contributes to the shape of our cells, but also acts as roads inside of cells. Okay, Grace, let me see if I can go back. Oh yeah, we're working on that. Ooh, there it goes. Microtubule. Actin and myofilament, that's good. Oh, did I not? Oh, I did the wrong one. All right, hold on. Let me do this. It should be, wait, I'm gonna stop. I'm gonna make y'all do it again. And I'm gonna make y'all do multiple choice. No, go back. All right, thank y'all. Okay, so I'll give y'all till 1 minute. Okay, we're past 1 minute. All right, so what did y'all say? Here, no, this one. Okay, so C, it should have been C. It should have been C. Tubulin are intermediate filaments. The two that could have been, right, the shape of the cells, we're talking about actin and tubulin here. The other one, actin here, doesn't act as a road. It acts as a skeleton, and it also functions a lot in contraction of muscles, which is what the very last section in this chapter is about, but it's not a road. When you think of a road, there's a really cool, I'm gonna have to share it with y'all, there's this really cool animation of a myosin on an intermediate filament, which is tubulin. And it's got like an attachment to a vacuole or some kind of cellular compartment. And it swings around and it uses its motor heads to drag the cellular compartment along with it wherever it wants to go. And so tubulin can act as those roads. So it can help to move and shuttle different cellular components to different parts of the cell. But actin doesn't do that. It's just tubulin. Okay. All right. So this is a major structural component of connective tissue, such as bones and tendons. So give y'all a minute. And remember, this question is not asking you inside the cell, right? Bones and tendons are what? Extracellular. Extracellular. Okay. So which one Did we pick? Good. Collagen. Collagen is super strong. It is super, super, super insanely strong. And it is a polymer that sits in the extracellular matrix and maintains structure and provides tensile strength to bones. This is why bones are so strong, because they have so much collagen in them. All right, so we said collagen. Okay, here. Blank are a type of cell signaling protein that reside on cell membranes. They include the G protein coupled receptors and receptor tyrosine kinases. This one should be pretty easy. Okay. So what did we say? Good. These should be membrane receptors, right? It resides on the cell membrane. That should be kind of your big giveaway. But you do need to know the different types of those receptors, and we'll talk about those. Okay, did I mark it correct? Yes. All right, so these are soluble cell signaling proteins that function as transcription factors to alter gene expression. Soluble in water. Soluble in water. So that's a good question. So let me ask you this while you're answering, right? If you have a soluble receptor and its function is to act as a transcription factor, does it need to be embedded in the nuclear membrane? Okay, so nuclear receptors, right? So does this nuclear receptor, does it need to be embedded into a membrane? No. Why not? What are you trying to do with it? What's the point of having a transcription factor? Change gene expression. Where's gene expression? On the DNA, right? It's not on the membrane of the nucleus. If you want to change expression of a particular gene, you're going to have a protein that can go in and actually bind to the DNA and alter its transcription in some way, shape, or form. It might alter the DNA. It might just increase the affinity of the polymerase for the DNA. There are a lot of different things that it could do to facilitate that function. But it does not need to be a receptor that is sitting on a membrane. These typically are not. They're soluble. They're floating around in the nucleus, doing their job. All right, so we said nuclear receptors. I gave you that. Okay, now, here's another type of signaling protein, but these function as molecular switches. So they undergo conformational changes in response to incoming signals in order to transmit or propagate that signal. we're only getting 47. I think 50 people checked in. I think we're only getting 47. So 5 seconds. Ooh, about 48. 49. Woohoo. Okay, so what did you say? You should have said intracellular signaling proteins. So these molecular switches are not sitting on the membrane. What do you have sitting on the membrane? This is some kind of membrane receptor, right? When the membrane receptor binds to its target, it's going to undergo some kind of a conformational change. And then there's going to be a protein that's associated with that membrane receptor. And when that membrane receptor undergoes a conformational change, now this protein, which is our intracellular signaling protein, will undergo a conformation. And maybe it gets spiky or something. I don't know, right? But it's going to change. And that's going to then propagate a signal throughout the cell so that you can have a receptor that sits on the outside of the cell that monitors what's going on out there. But then you can also get that signal to go all the way through the cell. So you have to be able to transmit that signal. All right. So now, globular transport proteins. This is where it gets a little more complicated, right? So y'all know that when a woman's pregnant, right? That everybody says, you know, okay, where's the baby getting oxygen from? The baby's getting oxygen from mom's blood. How does that work? How is it that mom can provide the oxygen for this developing fetus? So you should have watched the animations and the lecture on this. So I'm going to let you choose, and then we'll talk about it. I'm not sure what you mean, Ross. Give y'all another 20 seconds. Okay, so let's talk about what y'all say. Ooh, good, y'all said C, good. All right, let's talk about it. A, adult and fetal hemoglobin molecules are exactly the same. Are they exactly the same? No. There are some subunits that are different in fetal versus adult. Fetal hemoglobin proteins do not bind oxygen. If it's hemoglobin, it binds oxygen. So that's a big no, right? Okay, adult beta subunit of hemoglobin is replaced by a special hemoglobin subunit in which histidine 143 is replaced with serine 143, among other changes. Even if you didn't know that those were the residue changes, that one's a possibility, right? Look at D, more than 90% of a newborn human baby's hemoglobin exists in the adult form by the time the newborn is 1 month old. It's actually 6 months old. It takes 6 months, but that's not really going to explain how mom's blood oxygenates the baby's hemoglobin, right? So what's so special about this hemoglobin subunit that's in the fetal blood? Why does it work? What's different about the fetal hemoglobin versus the adult hemoglobin other than the residue changes? Yes, there are residue changes. Okay, yes. So Hannah's partially right, Ross is partially right. So Hannah said earlier that it's the umbilical cord. So the umbilical cord delivers blood to the baby, right? But that's adult red blood cells. But what happens when the adult red blood cells get in contact with the fetal red blood cells? The fetal hemoglobin is going to actually steal the oxygen from the maternal hemoglobin. How does it do that? How does it steal it? It doesn't bind BMP. No, bisphosphate, BGP. So the point is that it doesn't bind an inhibitor. The fetal hemoglobin will not bind an inhibitor, right? And because it doesn't bind an inhibitor, it will bind with a tighter affinity to that oxygen. So it'll basically steal away the oxygen because the inhibitor will prevent maternal blood from picking up our oxygen, whereas the fetal blood will be able to pick it up. So that's why we have the transfer of oxygen from maternal blood to fetal blood. It has to do with the affinity of the fetal hemoglobin versus the affinity of the maternal hemoglobin. So it's all about affinity. That was a big, I know that the second section was really all about hemoglobin, but really you learned a lot about affinity. So the attraction of molecules to one another, right? And affinity curves and things like that. So you have to be able to take that information about affinity and apply it to a new situation. So that's what I'm going to ask you to do next. I'm going to tell you that we have, I think I did, yeah, check that one. Okay, that we have two proteins. I'm going to give you a situation. You have ligand X and you have ligand Y, and they're going to bind to protein Z, right? So this is like oxygen binding to hemoglobin. Doesn't matter what the ligand and the proteins are, right? Now, I want you to analyze some data. There's an interaction of ligand X with protein Z, and it has a KD of 0.2 millimolar, while the interaction of ligand Y with protein Z has a KD of 0.6 millimolar. Y'all remind me, what is the difference between a KD and a KA? Before you vote in, what is the difference between KA and KD? One's association and one is dissociation. So if I say X plus Z goes to X bound to Z, right? How would I write my dissociation? How would I write my KD for that reaction? Whoa, sorry. Okay, so if I want to write my dissociation, I need, remember anytime you're doing a K, K is equal to products over reactants, right? But KD, I'm writing it in reverse. So I have to put my reactants on top. A and B say the same thing. Oh, A and B do have this. No, they don't. Yes, they do. Sorry, y'all. Well, then at least you can eliminate some, right? X, if I could write, and Z, right, over XZ. So how's that different from my KA? It's flipped. XZ over, and I'm writing all over my thing, X and Z individually, right? So it's the same reaction, but you're flip-flopping the reactants and the products to express an equilibrium expression for the dissociation, the two things coming apart, than you are for the association, which is two things coming together, okay? Dang it. So make this one a lower affinity. A lower affinity. Okay. All right. So if the interaction with X and Z is a KD of 0.2, but Y and Z is a KD of 0.6. Oh, I did. Yeah, that's right. That'll fix it. To move this. I need to go back and fix that. So just make sure you make that correction on B. Give you all 10 more seconds. Okay, come on, y'all, check in. All right. Okay, so what did you say? So what you should have said is that is a higher affinity. So if we look at our KD values, the lower the KD value, the more tightly the two molecules are held together, right? So if ligand X has a higher affinity for protein Z than ligand Y, then its KD should be what? You should have a smaller KD. What could you tell me about Ka? How can I flip that on its head? A higher Ka, right? So you have to be able to switch back and forth in your mind between the two, right? B would have been, if I had done it correctly, the opposite, right? C, KD is the dissociation constant, that's true, which is the equilibrium constant that expresses the protein ligand complex concentration as the numerator in the equilibrium expression. Is that what we had? Is the complex in the numerator? Here's the complex. Is it in the numerator? No, it should be in the denominator. D-E-N. All right, D, the larger the KD value is, the larger the Ka value will be. No, they have what kind of a relationship? Not an equal relationship, but an inverse relationship. So the larger the KD, the smaller the Ka, or vice versa. You could have changed it either way, right? Okay. I got one more on this one. And this one, I'm not going to do multiple choice. I want you to do short answer, and then I want you to do, you know, like A, comma B, comma C, comma D, whatever you got. So this one's about sickle cell anemia. So remember when you have mutations, right? In a particular protein, you're going to get sickle cell anemia. So I want you to read your statements, and I want you to tell me which of all of these statements are true. Could be more than one. And I'll give you a few minutes to answer that one because I want you to read through them all. Okay, I'll give y'all 5 more seconds. Okay. Put something in. All right. So when you were looking at this, did you go, oh my God, she's going to make us remember which mutations in the gene control for sickle cell anemia? You don't have to memorize that. You don't have to memorize that. So look at the first one, a mutation of residue 6 of hemoglobin from glutamate to isoleucine. Tell me about glutamate and isoleucine. One's charged, one's not. That's a big difference. That's a very, very big difference. You are likely to see a mutation of this sort lead to some kind of a phenotype. Okay, so I would say would likely. And you see how I'm covering my bases, right? Would likely. Does it necessarily? No. But you should be able to look at those residues. This is why it was really, really important for y'all to remember those amino acid side chains, so that you remember that. Okay. An effective method of treatment may include inducing the adult overexpression of the gene that codes for the gamma subunit of fetal hemoglobin. What do we know about fetal hemoglobin? It binds more oxygen. So these people who have sickle cell anemia, when they have an attack, their attack occurs when they're in low levels of O2. If they're in low levels of O2, that's when the cells start to sickle. So if you can keep the same amount of oxygen in the cell, but have more hemoglobin that's bound to that oxygen, are you necessarily going to induce that sickle shape and then have an attack? No, right? So if they were carrying around just fetal blood, they would be less likely to have a sickle cell attack anyway. So if we could overexpress that, that's potentially a treatment. So that one's yes, right? Okay, individuals are born with this genetic disease and symptoms can usually be diagnosed in newborns. True or false? What did we just say about newborns? They have the gamma subunit. If they have the gamma subunit, they're going to have lots of oxygen. So while it is a genetic mutation and they have the mutation, they're not having a phenotype because they have that gamma subunit, and that gamma subunit is preventing them from having an attack. So usually we don't see it until the children get older, and then they start to have a phenotype. Okay, so this one's incorrect. A mutation of residue 6 of hemoglobin from glutamate to aspartic acid would likely not cause sickle cell symptoms. Tell me about glutamate and aspartic acid. It's only 1 carbon difference. Probably not going to have an effect. Right? You go from leucine to isoleucine. You make small changes in the side chains, you're probably not going to have an effect, right? So that's true. A mutation where we go from glutamate to aspartic acid would likely not cause symptoms. So what you should have said was A, B, and D. A, B, and D. A, B, and D. Tell me if I missed one, y'all. No, I think that was it. A, B, and D. Oh, I guess that was a tricky one. So you don't have to go in and memorize what are the residue changes from the wild type to a diseased version of any of the diseases we talk about. You don't have to memorize that. What you have to know are the structures of the amino acid residues and then how those mutations might affect overall protein structure. And then you can apply that to any disease, to any mutation in the DNA. That's why it's important for you to know those structures, okay? All right, so what do we have? 5 minutes? Oh, I want us to have a little bit more time, but that's okay. So I'm going to let you, for the rest of the class, work on looking at your chapter 6 long iClicker questions. I want you to kind of look at that first one while you're here, just so that you understand what you're looking at with that alignment of genes, because we really haven't looked at that way before. So read through it, make sure you understand what that question is asking for before you walk out the door, but I'm going to let you go. Okay? Any questions? Once you're sure you're good on that question, you're good to go. What is the what? The single dot means that it's actually an asterisk. So watch, let me pull it up so y'all can see. The single one is actually an asterisk. Okay, so the single one, like if you look, right, there's nothing, nothing, and then the first one is a dot, but it's not really a dot, it's an asterisk. So the asterisk means that it's highly conserved. It's like 100% conservation. Then you have 2 dots. Oh, here you have less sequence homology. It's not as good as 2 dots. Yeah, it's not as good. Like there is some, but it's not as good. Alanine to valine. That's a bigger change than, let's see, what are some other ones? We have phenylalanine, leucine. So it's just about degree of homology. Okay, was anybody able to load up the long clicker questions? Let me go back and check and make sure you did. Hannah, why don't you close your, like, your Chrome or whatever browser you're using for that, and then restart it and see if that'll work. Thank you, Ross. Everybody else was able to access it? All right, so we got one more class period, and then we have another exam. It's getting close. All right, if y'all are good to go. If you don't have any questions on it, you're good to go. And I'm still in the chat if anybody needs. See who's still on. I'm going to stop the recording, but I'm going to stay on.